Daisy,
Hi, I'm David.
and we're the MSA Crusaders.
Welcome to another episode of MSA Crusaders. Today we're speaking with a very special guest that we waited to hear from. He was on our show about 2 years ago. Here we have Professor and Chair of the Department of Neurology at the University of Arkansas, Dr. Dhall.
Welcome, Dr. Dhall.
Well, thank you, David. Thank you, Daisy, for having me back.
It's a pleasure. We had a great conversation with you before, and we're happy that you could join us again to expand on another topic of research for patients with MSA.
Our viewers loved you before.
Yes.
You remained our number one episode for 2 years.
All right, let's see if we can better that.
Yeah, apparently we have some success with some of these clinical trials. So really the only question we have is you're part of some new revitalized research on MSA clinical trials, and we wanted to know there are very similar clinical trials to what you're doing from what I understand, from what I read. We would like to better understand what you're doing and how it differs from the other clinical trials that other pharmaceutical companies are pursuing.
All right, David. So, I'll kind of start with the overall foundation of how clinical research is done for bringing a new drug to the market for an LS. The process is very similar when it comes to developing drugs for all sorts of disorders, including degenerative disorders like MSA. We'll speak briefly about what some of the implications are for if a drug is being fast-tracked, if there's a breakthrough designation of the drug. But really, drug development requires the assessment of a medicine in a toxicology packet in animals before it can ever come to human trials. And then in people, trials are done in multiple different phases, with phase 1 research being first in human studies of a medicine to try and understand, usually in normal volunteers, whether the medicine is going to be safe, what the blood levels or other types of target levels that are achieved with that medicine. And then once the drug is through phase 1, it's typically studied in phase 2 studies, looking again at a longer-term safety signal of the medicine. Sometimes phase 2s, or late phase 2s, or what are called phase 2Bs, include effectiveness signals. Is the medicine going to improve the disease state in some way? And then usually the FDA requires an even larger studies called phase 3, where the group of people that are exposed, or the time period that they're exposed to the medicine, is even larger, so we could look at both the effectiveness and safety. So phase 2s are primarily looking at safety of a drug. Phase 3s are primarily looking at effectiveness of the drug. And for some things, like if there's a common infection, if we wanted to bring a drug for the common cold to the market, common things being easy to recruit for, we could do the sequential development of medicines rather quickly. For rare diseases, this is a challenge, because to get a group of, say, 150 people with multiple systems atrophy, we would need to bring on board many, many, many sites within the United States, and sometimes outside of the United States, so that we could get enough people who meet the very strict criteria for being a candidate for the study. I know, David, you and Daisy can share your personal experiences with trying to be a part of a clinical trials protocol, something that many of our patients, once they understand that that is a part of developing new future treatments for the entire group of people affected by MSA. They often are interested in pursuing those, but there are limitations based on the eligibility for the protocol that make it very difficult for people to participate in clinical trials. So because of those reasons, the researchers that are developing the drug have to kind of pool our resources. Usually development of the drug becomes a lot more expensive. The FDA also wants their regulatory authorities in the US, outside of the US, they want to look at long-term safety signals of the medicine to make sure there's no significant harm coming in the long term, or we understand what the risk calculus is for the drug. And all of that means that taking a medicine from where we've identified it in a petri dish or in a research lab through clinical trials development to the pharmacy where our patients can get that treatment often is a period of 7, 8, 10 years. Um, so that's kind of the background in which we work, in which a researcher like me works. I don't work with pipettes and petri dishes. My main interest is in finding treatments for people that I'm taking care of in the clinic. And so it is actually living through this experience of taking a drug through various phases of development. Also, at our center, we don't do phase 1 or first in human trials. They typically require people to stay in the hospital for periods of time, and that is an even more resource intensive task. Plus, most of them are in normal, healthy volunteers, college students. And, I think my first responsibility is for my patients or in imminent need of these therapies. So we start to get involved in phase 2 or later development of medicines. You brought up about research that is active at our center and other research projects that are active at multiple sites across the country for finding new treatments for multiple systems atrophy. Now, I'll share that our interest is not just in developing new treatments. I think it starts with identifying, understanding multiple system atrophy at various levels. On the patient-facing side, it is recognizing multiple systems atrophy. I think both of you have lived through where the diagnosis was not made clearly for a long period of time, and that's if you're declining for that 6 months, year, often longer, year and a half to 2 years. Like I think for Daisy, it was 2 years before we had a clear diagnosis. That is lost opportunity to intervene, both for helping manage symptoms and for doing something to stop the decline. And so better identification of multiple systems atrophy as a disease state, finding biological markers that tell us, are we certain about the diagnosis, and do we have certainty also that we can reliably get you into a clinical trial to change the progression? Because trials are the treatment is directed at specific targets. And if we're not even playing in the same sandbox, that's a problem for somebody coming into a clinical trial. And by the time there's high certainty that we're dealing with multiple systems atrophy, there's also been progression of the illness.
Yeah.
So, these are layered, kind of complex topics, but we remain committed to develop better what are called biological markers or diagnostics for multiple systems atrophy as well. And on the side of education, so I'm in a program where we train residents to understand Parkinsonian disorders in general and how to separate MSA from other disorders. But people have been in practice in my state. Again, it's or across the United States. It is my imperative as an educator to make sure they understand what's the current awareness about recognizing MSA. So some of what we do is working with organizations, including the MSA Coalition and the Cure PSP Foundation, in developing better ways for providers, for doctors, for nurse practitioners, for other people that our patients are going to come in contact with, like speech and language pathologists or physical therapists or other team members, to say, well, this is not Parkinson's disease, or this looks like multiple system atrophy. So the effort that we're engaged in is on multiple prongs. Education on recognizing MSA early. When it comes to specifically finding new treatments for multiple systems atrophy, the kind of most progress, the kind of longest thrust of development has been on a misfolded protein called alpha-synuclein. Alpha-synuclein is the shared kind of misfolded protein related damage that is a common thread between Parkinson's disease, multiple systems atrophy, and dementia with Lewy bodies. But in MSA, unlike in Parkinson's, the damage that alpha-synuclein causes is more extensive. And when we look at the under the microscope and we look at the brains of people with MSA, unlike in somebody with Parkinson's where alpha-synuclein clumps up in the nerve cells themselves, in MSA it clumps up in glia. And I think we talked briefly about it the last time. But we don't have the ability in a live individual to look at the actual clumping of alpha-synuclein at this time. There are tests, including PET scans, that are in development to see if we can find ways to separate MSA from Parkinson's disease. We rely on clinical features. There are criteria that the Movement Disorders Association has developed, to the Movement Disorders Society has developed to kind of recognize multiple systems atrophy with varying degrees of certainty early in the disease process. We rely on imaging markers. MRI scan has been quite useful in our understanding of early clinical changes in MSA. And all of those add up to our ability to get people into a clinical trial early. But as I said, so our main thrust has been, can we find a way to stop the cell-to-cell spread of misfolded alpha-synuclein, of this protein that kind of is a shared thread between Parkinson's, MSA, and disease. And so one of the trials that we were a part of that just closed, and a trial that is in the process of being opened here at UAMS, both are targeting misfolded alpha-synuclein through antibodies, kind of the your body's own defense mechanism against various pathogens, against viruses, bacteria, misfolded proteins like alpha-synuclein. The trial that we're going to be opening up soon is called MASCOT, MASCOT. And it is testing a specific antibody. The name is LUAF82422. When you hear that name, you know that it's not ready for prime time. There's nobody's going to be writing a prescription with that name. Hopefully, the market, it'll have a still hard to pronounce, but maybe easier to spell name than this one. So it's this particular antibody from a company called Lundbeck that is being tested in phase 3 trial. The previous testing of this, which since it's in phase 3 and phases are sequential, it means that it's gone through testing in phase 1 and phase 2 studies. The phase 2 trial that tested the same drug was the acronym for that was AMULET, AMULET. It was a randomized control trial with people being randomized to either getting this medicine or being on a placebo. And the medicine was deemed to be safe. The side effect profile was low. It has to be given as an IV infusion once every 4 weeks. And the trial was for a period of between 48 and 72 weeks. So close to a year and a half of use of the medicine. In phase 2s, typically safety is the primary goal of interest, making sure we're not making more problems, creating more issues for our patients. This phase, so it was a phase 2B late, phase 2 study, efficacy signal was also of interest. And the way they tried to look at effectiveness of this medicine was to see if a rating scale for MSA called UMSARS, Unified MSA Rating Scale, if the combined change in that rating scale was different between the two groups, suggesting that the group that was on the medicine progressed slower. And as a whole, the trial recruited about 60-something patients. And as a whole, the two groups did not seem to have a difference in progression of the UMSA rating scale, even though the medicine seemed like it was clearing misfolded alpha-synuclein reasonably well. But when we look at secondary analyses, what are called secondary analyses, which are analyses that we already defined we were going to do in the entire data set, but we're not going to be able to move the needle when it comes to FDA registration of the medicine.
Right.
It wants a pre-specified primary analysis to be positive for them to say, well, this looks promising, move it forward. But in secondary analyses, the individual parts, part 1 and part 2 of the UMSA rating scale, the Unified MSA rating scale, both showed a slower progression in the group that was on medicine compared to placebo. And then in the subset where MSA-related disability was lower, was lesser, there was an even larger effect size noticed on the changes in MSA rating. So promising results from AMULET, although would not count towards registration with the FDA. That's why the company is moving forward with continuing to pursue development of the medicine in this phase 2. The previous trial that was open from a company called Takeda Pharmaceuticals with us is closed, but the data is still being collected. And so the kind of nice thing is that we're trying to hedge our bets. We're trying to say, we need therapies for this. We're committed to, testing multiple different types of medicine simultaneously to see if something will work. Both of these are antibodies against alpha-synuclein, so medicines that are going to hoover up the misfolded protein and allow your body to clear it better.
Yeah.
But there are other ways of trying to slow the misfolding of alpha-synuclein. We could work on immunizing your body against alpha-synuclein so it can make its own antibodies, so you don't have to get antibodies from outside. We can have a reduced expression of alpha-synuclein through some pretty neat treatments called antisense oligonucleotides. Misfolded alpha-synuclein when it gathers up, it causes inflammation in the brain, and we could target that through different strategies. There are changes in the way your nerve cells and the glia handle misfolded proteins in general, and we could target that. So many of those are actually being explored. There is also movement on the stem cell side, which I think, David, you probably are able to share
Yes.
more information with the audience about that. And so, we're continuing to work on ways to slow the progression of multiple systems atrophy. In addition, we need better treatments, as both of you can attest to, and what's called an investigator-initiated study that is funded very generously through support of our donors here. Somebody living with multiple systems atrophy. One of my partners, Dr. Vermani, is looking at the effects of medicines on speech changes in multiple systems atrophy, on swallowing changes in multiple systems atrophy, two major issues that make it difficult for people living with MSA to communicate, to make sure they're getting enough nourishment, to not have issues with choking or aspiration. And so and it's surprising how little. Many of us think that medicines that we currently have may improve speech and swallowing, but it is surprising how little we actually know about the effects of those. So we're trying to actually study it in a group of people with both MSA and with another condition called PSP.
PSP. Very. I was going to say, very happy that your group is, taking a former clinical trial that has had some success, right, and trying to reincarnate it to, change it to possibly us getting a flu shot for a cure. I was reading how, the goal is to have this whole alpha-nuclear protein digested out the body naturally. And, we just want to say thank you for picking up where others have left off, because this is really hope. I mean, last year there was no studies that were saying there was any progress. And now over the last year, year and a half, we're starting to see like you and your team with the mascot, we're starting to see more positive steps towards a therapeutic treatments and probably a cure in the short term.
We remain committed to finding new treatments and cures, and I use the royal we here. There's like a group of us spread across continents. In the US, there are several sites for mascot, for example. In Europe, there are sites in Canada. So many of us are answerable directly to, people like you and Daisy Page, where, they're living with the disease, they're seeing the clock ticking, and they're looking at us for answers. And we're committed to trying to, search those answers, hopefully bring better treatments to slow progression. I think the first step towards restoring the nervous system is to stop this fire from kind of continuing to simmer and cause more and more damage. That's when we can start to think about restoration.
Yeah, I agree, because Daisy and I spoke about this before, and I'm sure with our families, like myself personally, if we stopped right now, we could stop the progression, I would be thrilled. I could still do a lot in my life. Daisy, yourself, nobody wants to die, right? But being able to at least slow it or stop it would be the greatest thing ever. So that's, kudos to you and your team for jobs. Well done. We have you on here just not for information purposes, but for people to see that what's happening behind the scenes and actually get them to also apply for clinical trials to be a part of it. Because without our listeners and watchers on YouTube and stuff like that, we have very few people that even know about clinical trials, or where they're at, what they're doing, the exclusion criteria for them. So, thank you for helping us spread the word about MSA in your specific clinical trial.
Thank you, David.
Linda can vouch for it. They were just not far enough along for DDNF, and I was too far along DDNF.
Yeah.
Right. And I think that's one of the important things about this is we're, a mentor of mine says, we're not going to catch fish unless you go fishing.
Yes.
For our patients, it is a difficult decision often, because being in a clinical trial requires you to follow very pre-specified, specific, visit schedules, and you're taking a risk on a medication that we don't have the entire safety data set on. You're taking this risk in advance with the hope that it's gonna change the disease trajectory. And when we find out that the trials on the whole, for example, Amulet was negative, it is disappointing. But think about the group that did participate early in it. We're seeing that even though on the whole, the entire group, we couldn't see a difference between those who got the medicine versus those who didn't. In the group that were, had lower level of disability to start with, their change over the year appears to be significantly different. That definitely gives us hope. You're taking a chance. You may, go to a site and they may say, well, we've looked and unfortunately you don't qualify for the study because of such and such a reason. Those inclusion or exclusion criteria strictly are for safety purposes. They're also for the purposes of the FDA being able to get a clean data set. And so at times it may be disappointing you weren't able to be a part of the study, but at sites like ours, our job is to make sure you get informed about your disease as well. So we're going to make sure that there's something positive that comes out of you raising your hand and saying, this is something I'm willing to do.
And that's much appreciated too, by the way. Very much so.
Oh, no, thank you so much for taking time with us today. We really, really appreciate it.
Well,
Absolutely.
it's my pleasure, and good to see you guys again. And I hope this is well received as well.
Because we all are MSA Crusaders.
We want to thank you again, Dr. Dhall.
My pleasure.